map very well with what Charles Goodnight talked about. That we have a lot in common in that we come from what was called the Chicago School of Quantitative Genetics. I was at Northwestern, not Chicago, but Mike Wade, Steve Arnold, and Russ Landy influenced a lot of people, including myself and Charles. So I say I rarely hear everything described so perfectly correctly, the right way to think of things, which is the Sewell right way of thinking things. It comes out of the University of Chicago. I'm going to talk about quantitative trait locus mapping and how we can use that to address questions of the genetic architecture of complex traits. And today I'm just going to go through some basics of mapping and some of the experimental results we've gotten over the past 20 years or so. Tomorrow I will talk about, or next lecture will be about epistasis. And I have my own take on epistasis, a little bit different from the way Charles described it, but we end up in exactly the same spot. So it's all really the same, but you have here it coming from a different direction. The graphs he showed are all, there's nothing inconsistent between what we're saying. So if something seems inconsistent, please ask about it. And then the next will be about pleiotropy and how pleiotropy can interact with the genetic architecture, epistasis and dominance, and the importance of both epistasis and these, that should be working now.
You have 24 hours worth of...
24 hours I got now.
Yeah. No.
Charles, I didn't see that long. So excited. That went by quickly.
I don't know. Maybe we got out of control.
So we'll talk here about quantitative trait locus mapping strategies. So here we're trying to discover and measure the effects of the individual genes that underlie complex traits and that underlie evolutionary change, and morphology is what I'm mostly interested in, but morphology, behavior, physiology, all sorts of different traits. They're complex traits, by which I mean they're affected by many genes and the environment, and it's difficult in a distribution to find the effects of an individual gene. As Charles pointed out yesterday when he kept adding loci in to the effects, and he ended up with a normal distribution of phenotypic traits. So that's determining the genetic architecture of complex traits, additivity, dominance, epistasis, pleiotropy. Another purpose, and the one that actually the U.S. federal government will fund, is to determine the molecular basis of quantitative differences in complex diseases. For this, you have to be able to name the gene. For the evolutionary purpose, you don't need to name the gene. You just have to specify the effect. So if you're interested in doing trying to pull apart the genetic architecture of a complex trait, you start by choosing, in experimental studies, genetically differentiated populations to cross. My work has been in house mice, and in using originally the inbred strains of house mice. These are very convenient for such experiments. We now have the full sequences of about 20 strains of the mice. You have a wide array of phenotypes available to you. It's just a matter of picking the right pairs of strains for your purpose. So when we first started, we looked through the literature. We did some preliminary testing, and we looked up in the literature the phenotypes of our strains. And you can find out phenotypes for a wide variety of traits at that website that's posted here, the Phenome Project website. We were interested in studying growth. So we looked for two strains that would grow differently. When you look at these strains, you want these to be very different phenotypically so that they have a high broad sense heritability and the likelihood that many genes, not just one or two, are likely responsible for the strain differences. If it's just one or two genes, it's not a very good mimic of a natural situation. So here's a list of a lot of strains of mice. You can see that there are different colors on the list, and the colors represent different parts of the mouse lineage. Really, the house mouse, the inbred strains of house mice, phylogeny is mostly a bush. But there are some relationships down near the base of the bush, and those are shown in the different colors. So often you will pick strains from different groupings to make sure they're genetically diverse. And since we were going to look at growth, it turned out there were two strains. One's called LG, up there at the top right, that's large. And the other one, SM, down at the bottom left, is small. So we figured large and small might differ in their growth. And that would be a logical choice. So the two extremes of body size in the inbred strains of mice. So that's why we chose them. Here's a picture of the mice, and it's a large female mouse and a small male mouse. That's their strain names, not just their body sizes. So you can see that they're fairly different in size. The white female mouse, much bigger than the black male mouse. We did the experimental cross just one way because of a big fire at Jackson Laboratories up in Bar Harbor, Maine. Their breeding colonies burned, and they had to reconstitute many strains, including small, from the freezer, frozen embryos. And as we all know, females are worth a lot on their own. One male's as good as another. So they would sell males, but they wouldn't sell females because they needed them to build their colony back up. They only needed one male around, really, to produce the colony. So they would sell them off, and thus our strategy was decided for us by the fire. The size difference is even bigger than it shows in the picture because this is a female large and a male small. The female small would be even smaller than the male would. There's sexual dimorphism in the strains. So these two strains have a long history. They were begun in the 1930s, and they come from different origins. The small mouse on your left was an experiment by a guy named MacArthur. And MacArthur took 7 different strains of mice and he bred them all together. He gave the names of the strains, but we don't know what that really means anymore in terms of modern names. And he got a diverse population. He then selected up and down. And you can see the selection results here. Male and female, small male and female. He had a large strain as well. It would have been ideal for us to use that strain, but it's dead. It's gone extinct. So we used this small strain. You can see that there was a strong response to selection over time. You've never heard of MacArthur and Wilson? Is that a familiar? Have you had any ecology, island biogeography? This is that MacArthur's father, and they did field research together for many years. But the father died relatively young. Goodell was working at an agricultural research station funded by the Rockefeller Foundation. And now it's by Williams College in Massachusetts. And he was going to try to grow a mouse as big as he could get. he wanted to see how big he could make the mouse. Could he make it as big as a rat? Could he make it as big as a sheep? A cow? You know, what were the limits on this?
Elephant.
So he got a bunch of mice in a box, and he would pick out the big ones. There's no mathematical selection strategy or anything like that involved. He just picked out the big ones. You see in the slide here a picture that comes from a little film of him with the mice. The mouse at the top is represents the size of the first generation, and then as you go down, it's the size each year during the early part of the selection. You can see the response to selection for the Goodell large white mice at the bottom right there. What's fun in the video, which I could not get a full copy of, is that the mice are all alive and they're all trying to jump off the podium constantly. So he's picking up mice, putting them back on. Anyway, he failed to get a mouse as big as a rat. That's just life. You could see that there was a very strong response to selection for some 40 generations, and then the heritable variation in the population was used up. And you can then see that the body size pretty much stayed the same, even though there was selection going on. However, this large strain has thrown a lot of mutants over time. A lot of the pygmy style or small mouse mutants come out of this line because they're really obvious when you're working with the mice that, oh, this one's really small. It's smaller than its brothers and sisters, you know, and that's because it has a new mutation. So small mutations showed up in this line and then were fixed. So large and small, first part of the 20th century. The mid part of the 20th century, they went to Jackson Labs, where they've been inbred since. And we started using them about 1995 or 1994. So the basic experiment to do mapping is to take two strains, two inbred strains preferentially. We have the large and the small. And you perform what's called an intercross. You make the large to the small. You don't randomly make these large and small animals. You only make large with small. And in doing so, you produce these F1 hybrid animals, each carrying a chromosome that's large and another chromosome that's small. These F1 hybrid animals are all genetically identical to one another. Right? They have the same genomes. This non-random mating actually has an important effect for the gene mapping. It does two things. The just crossing them and not randomly mating itself makes each chromosome independent of one another. Each chromosome is inherited independently in linkage equilibrium after this first generation of mating. Whereas normally it would take about 7 generations of random mating to eliminate linkage disequilibrium between two strains like this. The other thing it does, which is convenient for the mapping and the cost of the mapping, is that while it eliminates linkage disequilibrium between chromosomes, it increases it on chromosomes. And that's an advantage because we're going to mark the chromosomes with markers. And we're going to tell where genes are from their correlation with the markers. The correlation between the gene that's actually affecting the growth and the marker is the linkage disequilibrium between them. So it takes less markers to fill out a map in an F2 than it would in another population, which makes it cheaper. Back when we first started this, that was important because there were so few markers. But now that there are, 17 million SNPs known in mouse inbred strains, that's become less of an issue. But the money is still an issue. So we take the two strains, cross them, and then take that hybrid. We make the males to the females. I work in St. Louis in Missouri, and you're only allowed to do that. You can't do anything else with males and females. Pair them that way. And there will be recombinations in those F1 parents. So each F2 animal is a genetically independent recombination of the parent strains. So even though they're born in a litter, the animals within a SIV ship are genetically unrelated. And they're as related to animals from other SIV ships as they are to their own SIVs. This is because of the breeding design, that everybody has genetically the same parents, the F1 hybrid. So since everybody has the same parents, you can either think of these as one big group of sieves, or as they are within its own universe, they are the genetically independent individuals. So you create a lot of these. And basically the QTL mapping that you do is an association between the phenotype, the growth or body size or whatever, and the genotype. So here's an example of a region that might be a QTL. And you can see that the first two individuals here are small homozygotes, because they carry two alleles that arose from the small parent. We have next to it two heterozygotes that got one allele in that region comes from large and one comes from small. And then finally a large homozygote on the far right. So you correlate these differences in genotypes with differences in the characters that you've measured. And that's really all there is to this gene mapping. So it's kind of simple-minded. You get segregation of genotypes, a Mendelian segregation of genotypes, and you correlate that with the segregation of phenotypes. So these strains, large and small, were first worked on separately from their experiments that generated them by C.K. Kai in the 1950s and 60s. And he was interested in complex trait genetics, and he was trying to identify loci that were responsible for body size differences between large and small. He did about 10-15 years of experiments on this and did manage to find a body size locus in the mid portion of chromosome 7. But he didn't have genetic markers, the things that we have today. So it took him 10 years to do what it took us a year to do. And we were able to actually identify smaller parts of a chromosome than he could. So molecular biology actually became useful in population biology when we got all this molecular variation available to us. So you see up there at the top, we have the two parent strains, large and small. Their body weights at 60 days, which was the selection criterion. You can see that the F1 falls right in between the two parents. So the cross is, say, additive. The F1 hybrid is midway between the two parents. The dashed line is midway, and the straight line, solid line, is the mean. And then you take those F1s and you intercross them, and that produces the F2, shown with the hatching in the center of the distribution of the bottom panel. We also did back crosses, and you can see the back crosses go halfway back to the parents, and it's all very additive. When you look at the number of genes involved, using what's called the effective number of genes, it's something Sewell Wright made up. This cross for body size was supposed to have at least 11 genes responsible for the difference between large and small. That's very unusual to get such a high number. It's almost always an underestimate. And a lot of times when you do this, you find 3, 4, 11 was one of the highest numbers that has been found for such things. So we thought it would be a good model for evolution and genetics of complex traits. Here you see our own F2 distribution of body weights, and you can see it goes all the way from 16 grams up to 44 grams. The small mouse is about 18 to 20 grams, and the large mouse is about 38 to 40. Here you can see it's a nice normal distribution like you'd expect when there are many genes affecting the trait. We collect phenotypes on them, and since we were interested in growth, we would weigh the animals weekly. And I know I need to get a new slide of this. This is kind of old. This is a portable computer, and it does not have a hard drive.
We remember that.
You know, it's no hard drive at all in there. We did have some trouble with it. You can see there's not a plastic covering it. Now there is a plastic covering it. The mouse leavings, mouse turds got stuck in the keyboard, and one day the escape key got stuck down, and the student who was taking the measurements couldn't get the computer to operate, and I had to come in, and sure enough, it was. It was sticky. We always write down the answer, but it also goes directly into the computer. Each animal weighed 8 times within a matter of seconds, and the average of those 8 weights taken as the animal's score. But we also do write everything down because I'm old-fashioned. It also, sometimes the computer can get crazy, and it's a check on that. When the mice reach their term and are going to be sacrificed, we do a necropsy. A necropsy is an autopsy done on something that's not human. This is a necropsy. Dr. Pericato, by the way, is the fastest necropsy person that we ever had working with us. Even faster than Susan. Susan still admits it. So. We would open up the animal and take out the liver, heart, spleen, and kidneys and weigh them. So in addition to having total body weight, we would have the weight of these organs. We would also weigh fat pads, and the 4 fat pads are listed over on the left there. There's a reproductive fat pad, which in males is epididymal with the epididymis. Females, it's parametrial because it runs along the uterus, along the uterine tubes. Humans don't have that fat pad, but it's the biggest fat pad in a mouse, and the place where they usually are storing their fat. The blood from that fat pad drains into the cable system, means it does not go through the liver first. The mesenteric fat, the 4th one, is the fat in the mesentery between the gut and the body wall. This is the fat that does drain into the liver, and physiologically has different properties than reproductive fat pad. There's also fat around the kidney and fat in the inguinal region, which is where the leg meets the trunk. So that was the reproductive fat pad. You gotta look quickly. That's the kidney fat pads, the inguinal, and the mesenteric. So all those are getting weighed too. Then the carcasses turn into skeletons. So we've done a lot of work on bone morphology and bone structure, biomechanical properties of the bone, and also the size and shape of the different parts of the skeleton. So you'll hear me mention humerus length today and things like that. So far we have about 5,000 skeletons, but we have freezers still full of carcasses to be turned into skeletons. They have to be skinned, dried, and then fed to domestic beetles. You eat the flesh off of them, and then you have a skeleton. We often had trouble with our domestic beetles and getting them to really want to eat until Arthur Porto came to the lab from University of Sao Paulo. And Arthur had the brilliant idea of getting, instead of going to a biological supply house to get the beetles, he bought them on eBay. And therefore they came from much better colonies than Ward's Scientific. And we're cheaper. So now it goes at great speed. From the tissues that we take out, typically from the liver, we will extract DNA and then use that DNA to score markers. Back in the old days, these were microsatellite markers, short repeat markers. And that's what we've done in our lab. But a few years ago, with Jason Wolf's help, we went back and re-extracted all the animals from all of our experiments, about 6,000 animals, and genotyped them using SNPs. The advantages of SNPs are many. The most important one to us was that it was cheaper. It cost between 4 and 25 cents to get a genotype. Whereas with the microsatellites, it was at least 50 cents a genotype. The time it took us to do the microsatellites could be years for a population doing 100 microsatellites and 500 animals. It would take at least a year. With SNPs, you send your DNA away somewhere and an Excel file comes back like a month or two later and you have all your genotyping done. So it doesn't pay to do what industry will do a lot cheaper. So we don't genotype so much in the lab anymore. The results are more complete. With the microsatellites, we would have about 7% that failed. Well, we'd have whole plates that would fail. But with plates that worked and everything was right, we'd still lose 7% of our genotypes. Whereas 99.8% of the SNP genotypes end up scored. And then it's also more accurate. There are fewer scoring errors. When we go beyond an F2 and in later generations we have families, so we could tell when there was a scoring error. Because you would have an individual who couldn't have come out of the parents. Or there was an inconsistency between parents and offspring in their genotypes. So you could tell there was an error. And the error rate is much, much lower with the SNPs. So all around, better data, more power for the analysis. So in the quantitative trait locus mapping, you're going to measure the phenotype, and you measure the genotype, and you correlate them. And with that correlation, you'll get a probability of obtaining a correlation that high even when there's no relationship between the genotype and phenotype. Just by chance, it's going to pop up. So you use minus log base 10 of that probability. It's called a LOD score. And that's plotted along here. So a 1 is a probability of 10%. A 2 is 1%. 3 is 0.1%. 4, 0.01%, and so on. So as the numbers go higher, the probability of obtaining such an effect with no gene present is lower and lower. You have to figure out a statistical threshold for significance. And this becomes an issue because when you're doing this test, we're going to go all across the genome and do these correlations. So you do hundreds, thousands of correlations. And even when there is nothing, some of them might be significant at a 5% level, even by chance. 5% of them would be significant even when there's nothing. So there's a correction done called the Bonferroni correction, which limits the probability of getting such a result over the entire set of results to 5%, instead of only at one spot, 5%. And in the F2 for our population, the thresholds are usually between 2 and 3 in an F2. So it's plotted here then in these log to the minus log to the base 10 of the probability of a gene affecting the phenotype across the chromosome. The y-axis is the chromosome, The x-axis is the log score. And you can see there are peaks on this that rise above the significance threshold. You pick the highest peak, which is there. And this is the position of the gene affecting body size in the mice. You also derive a confidence interval, and you can say that you're 95% sure that the gene having this effect is between the limits of the confidence interval. And then you can look and see what genes are in the confidence interval, and hopefully maybe decide that one of those genes actually is the physiological one responsible for the effect. And you might notice that there's a second peak on this one, right? Or maybe even a third. As we come down pretty far here, and then we're going up again. These little peaks are actually an artifact of the statistical method and don't mean much separately. There would be a second peak here probably. It's probably far enough away from the first one that it's an independent second gene. And you would do a test whether there could be a second gene on the chromosome also affecting the trait. And then if you have a significant improvement in fit by putting the second gene in, then you declare yourself 2 genes on the chromosome. So we did something really unnatural. We replicated our experiment. It's always a dangerous thing to do. So we created 500 F2s and mapped, and the stars you see are for adult body weight, 10-week body weight, mapped in the first F2 intercross. So they're to the left of the chromosome. So you can see there were a whole bunch of genes. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 genes. Remember that according to the minimum number of genes, there should be 11. That's almost always an underestimate. So here we have 19. These are a cartoon of the mouse chromosomes with the genes on them. Thankfully, mice are, every chromosome is acrocentric. So you don't have to talk about long and short arms. It's just arms. I hate that people. I can never remember what P is supposed to be. It's supposed to be short because it's French for petit. P seems like it should be the long one, and Q is short. So we did the second experiment and mapped again. And there on the right-hand side of the chromosomes, you can see these marks here, the arrows. And those represent the positions found the second time. And you can see that many of the same positions occur here, 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 the middle one, chromosome 8, and so on. Many of them replicated. The ones that replicated were the ones that had the most evidence for them in the first place, in the first study. The second, but you can also see that there are places that did not replicate. Chromosome 1, chromosome 3, parts of chromosome 7. So we also didn't get all of them. Mm-hmm. And then you can see there's positions that were not mapped the first time that are mapped the second time, like the one on chromosome 17, the male and female QTL body weight on 16, and down here at the bottom, chromosome 13, top of chromosome 12. So we got some new ones too. So what would you expect with replication of the experiment? Well, you don't actually expect to get everything the same both times. And this is because we have many genes of small effect. And this is an example of what's called the Beavis effect, after a corn geneticist, Willie Beavis. And we're looking at 10 QTLs here that differ between our 2 strains for body size. The effect size is given here, the amount of what number of grams different in body weight. So they're all the same. But when you take a sample, some of the individual QTLs are going to be small, they'll be measured to have a smaller than their true effect. And actually, the other half will be larger than the true effect. If your threshold is well above the true effect size, only a few of the QTLs, only 2 of these 10 are picked up in your first experiment. And these others you don't see because their effects are too small. But you go back and you do the same experiment a second time and you get this result. Where now, again, half of them are above the true effect size and half are below, but it's 2 different loci that make it above the threshold by chance. So you can actually have a lot of differences between replicate experiments because of these statistical effects. You're going to pick up effects that are bigger than average, and you're going to think your effect is bigger than it really is. And when we did replicate, and we found it in the second experiment, often that value for the effect was smaller than it was in the first experiment. If you're doing human genetics and you do a whole genome scan, these GWAS scans where they have markers, million markers all over the genome, and they're doing this with humans, this is a major problem for human populations. We have a 90% power to pick up effects of like 0.2 standard deviations. In the human studies, their power is about 10%. So they find only 10% of the locations, the ones that by chance go above the threshold. And they miss 90% of what they're looking for. And then they talk about the missing heritability. It's not missing, used in fun. We also weighed the reproductive fat pad, and this shows the cartoons of the chromosomes again, with the reproductive fat pad loci marked on. There are 8 of them here. One thing to notice about them is that there's a lot of sex-specific results. That the fat pad has different genetic characteristics in the male and the female, different genes affecting the fat pad. We'll especially notice this adiposity 8, because that's going to come up some more. So when we look at these adiposity quantitative trait loci, the effects differ by sex. 4 only affecting males, 1 only affecting females, and only 3 affecting both sexes. All the QTLs have an effect also on overall body weight. What you might expect is the fat pad is part of the body weight. But we also measured the skeletal size, in this case the length of the tail. And you might expect some genes that would make an animal fatter would just make them bigger for everything. Their fat pad, their skeleton, just everything. But only 2 of the locations that affected adiposity also affected the length of the tail. So as we talk about genetic architecture, we're going to go through these questions. How many genes affect a trait? Well, as we've looked at different kinds of traits, the length of the long bones, the internal organs, the skull morphology, the morphology of the jaw, body weight, growth, you find, here it's between 10 and 30 genes. Actually, 30 is the most you could find because you're only breaking up you only have 1 generation of recombination. So it's only going to break up the chromosomes so much. So there may only be 40 locations in the genome you could specify. So basically we're finding genes everywhere in the genome affecting these traits. Secondly, how large are typical gene effects? So this is a plot of, that's a frequency distribution of the number of genes having phenotypic effects given on the x-axis. This is in terms of standard deviation units rather than grams, because we're putting results from a 1-week-old mouse together with results from a 10-week-old mouse. So we want to correct for the fact that there's different levels of variation in those different ages. So a comparable number to use is the standard deviation numbers. And you can see that the biggest effect we have is 0.5, a half of the standard deviation. None of these would look like Mendelian loci at all. They would be lost in the environmental variation if they were looked at singly, without markers. You can also see that there's a deficit over here on the left. That's just technical, because the effects over here are too small, so they rarely show up as significant. This is the additive effect. Some loci just have dominance and not additivity. Those are filling in here. But it's presumed that you can kind of see an exponential decline in the effect size over the effect sizes there, where an exponential decline from a lot of genes of relatively small effect to very few of larger effect. So if you extend this on up, once you get down to 0.01 or so, you would have maybe 70, 80 genes with that effect. Ones we can't see because they're too small. So this is always going to be an underestimate, and there will be a technical limit on this distribution of gene effects. You should realize that there is here. It probably does stick way above that. It's just you can't detect them as statistically significant. So do we find dominance? This is a distribution of the dominance to additive ratio. We'll talk more about the different kinds of dominance, but you can see basically when you're over on this scale, a minus number indicates that the small allele is dominant to the large allele. This side, the large allele is dominant to the small allele. Or in the most extreme cases, here and here, you have overdominance. The heterozygote is bigger than either homozygote. And underdominance, the heterozygote is smaller than either homozygote. Typically, the most common pattern that we find is codominance. But when there is dominance, we found that typically for size-related traits, large was dominant to small. The cases where there's overdominance, where the heterozygote is higher than either homozygote, were non-metric skeletal traits. That's like extra foramina for nerves, that is in the skull, things like that. Fluctuating asymmetry. And the early growth also showed a lot of overdominance. The heterozygotes grew faster in the first few weeks of life than either homozygote. Tell it's an old slide because it doesn't have other important traits that show overdominance, which would be maternal care and litter size. So we're looking here at the weight traits and the fat pad, and we took our F2 population and we randomly mated 200 pairs to produce an F3 population. This F3 population had 1,600 animals in it from then 200 SIP ships. So these F3 animals are no longer independent of each other. They're in groups, SIP ships. But being in groups, you can do ordinary quantitative genetics on them, and this shows the results for the first weekly weight, week 10 weight, and the fat pad. And the point of showing the slide is primarily to look at this, the cross-sex correlation. If the cross-sex correlation is 1, then genes have the same effects in males and females. And you can see that while this isn't 1, that's pretty close to 1 given error and measurement and things like that. So the effects on early growth are the same in males and females. Once you get to adult body size, though, the correlation is only 0.62. And if you square that, you could say that only 36% of the variation is shared between male weight and female weight. So there's a lot of sex specificity for body weight. And it's even more so for the fat pad weight. Only 16% of the variance shared between males and females. So in this population, different genes are affecting male and female weight, or different genes have opposite effects on male and female weight. So we're going to move on to some studies we've done more recently. And this involves, again, looking at body size and obesity. And here, though, we're adding in a diet. So the diet is a high or a low-fat diet. And basically, this is an illustration of the diet. I believe if you go down South Carlos Avenue, you could find something like this. There's yellow arches. So you feed this to some mice, and you feed a low-fat diet to other mice. These are the high and the low-fat diets. The high-fat diet is yellow, the low-fat diet is pink. They're both pelleted, but they're very different consistency. The people in the lab who have eaten these diets themselves don't like the low-fat diet. Don't like this one at all. The yellow one, you dip it in chocolate, you got, great stuff for a party. It's basically, a full stick of butter. Here's the statistics on that. The high-fat diet is 42% of the calories from fat. The low-fat diet, 15% of the calories from fat. However, overall, the two diets are isocaloric. So the same number of calories per gram in each diet. But one's got a lot of fat, and the other instead is making up for it with starch. So we went to an F16 generation of these mice. So I told you how we made an F3, but we kept randomly mating them on and on and on. They're at the F50 now. This is the F16. The reason you do this random mating and continue to do it for years and years is because you don't have anything else to do. So. It's hard to inbreed mice, easy to outbreed them. But each generation you accumulate more recombination so that you can map to smaller and smaller intervals in the genome with each generation. So we had 1,002 animals from 76 litters. We split the litters so that half of the litter got a high-fat diet and half of the litter got a low-fat diet. And we resulted, when you look at the diets and sexes, that basically we had 250 males on a low-fat diet, 250 females on low-fat, 250 males on high-fat, and 250 females on high-fat. So this is the same thing I showed before, where we had the F2 generation, and we looked at the QTL right here. You can tell the genotypes. If you go through, we used at least 60 pairs a generation, and we also tried to only take one male and one female from each litter to go to the next generation. If you eliminate variation in family size, you strongly retard genetic drift. So I was trying to have the least amount of inbreeding in this population for as long a time as we can. At the 50th generation now, they're about 8% inbred. But you can see that these little segments of red and blue are much smaller size in the F16 advanced intercross line than they are in the F2. So we'll be able to localize gene effects to a much smaller region. In fact, there's the factor of 8 involved. The F16 divide the number of generations by 2, and you get the accumulation of recombination. There's 8 times as much recombination here as there is here. Some of the mice do get fat. You know, if only good animal tried this, you wouldn't have been stuck, you know. Because this animal was about 85 grams. Animal from the same family on the low-fat diet would be maybe 35 grams. So probably more than half of this body weight is fat. When you dissect out the fat pads for an animal like this, more than half of the body weight will be in those fat pads, let alone the fat in the liver, the fat in the muscles, and the fat everywhere else in the body. They're greasy. They produce a lot of oil. You can see that it's not well groomed. You see the clumping of the hair there. So, female on a high-fat diet. So you go through and you do the mapping, and you do this for each chromosome. This is chromosome 2. And these are the LOD scores again. And the threshold is up here. The threshold goes up as you get further and further into the generations. And but you can see, unlike that first example slide I showed with the two big hills, and it was kind of hard to tell their wide ranges for them, these are really, it's the same chromosomes. This is really, ties it down to a very small interval. So we've got a much finer detail on it, and here you can see what showed up at first as a single location in an F2 is now divided into 3 different locations. So our QTL effects in the F2 are often the result of several genes together having similar effects. So you got the first result because you had 3 positive effects next to each other. With further recombination, those loci are divided up and become independent of one another. And you can identify the different positions. Is there a gene over here? Well, it sure looks like it. But it didn't pass the Bonferroni threshold. So you're not allowed to say that there's a gene there, even though there probably is. Bonferroni way overcorrects. It's just basically by chance that peak didn't get up as high as these peaks, probably by chance. And that's part of the reason you don't get to call it QTL. This one doesn't. It really gets over. So if we look at the number of QTLs we found 40 affecting fat pads. About 20 affecting the sum of the fat pads. Most genes for the reproductive fat pad, and there's 26. The inguinal fat pad at 8, 12 for the mesenteric, and 10 for the renal. If there really was no gene at all in the genome affecting these traits, I would expect these numbers to be 1, given the significance threshold that we have. So obviously, many more than the 1. It's possible that the reproductive fat pad, you find more genes, maybe more genes affected, but also it's a bigger fat pad, so there's less measurement error in it. So less measurement error means you have more accuracy and perhaps can map better as well. If we looked at the pleiotropic effects, whether these loci affected 1 fat depot, 2, 3, or all 4, using the Bonferroni criterion, this is the result you get, where the majority of the loci only affect a single fat pad. Another 25% do 2, 7.5% 3, and 10% 4. This is probably not a realistic look at the pleiotropic effects. That Bonferroni criterion is so severe that you are undoubtedly going to reject as significant things that are really there at an actually rather high level. The genetic correlations between the fat pads, the quantitative genetic correlations, are 0.85 and 0.9. They're very, very high. An indication that the same genes are affecting all 4 fat pads. So if we change from the strict Bonferroni thresholds to a situation where there is 1 fat pad that passes the Bonferroni threshold, and then we look at the other fat pads to see if they're significant at a local uncorrected 5% level, we get a totally different picture of what's going on. Instead now, about half of the loci affect all 4 fat pads. Another 25% affect 3, 22.52, and only 5% a single fat pad. This is more consistent with what we see from the quantitative genetics, and it's probably a more realistic view of the traits. Now we also are able to break down these effects into effects that occur in the whole population, and then effects that are restricted, either to animals on a high-fat diet or a low-fat diet. We expect a lot to be restricted by sex, only males, only females, and perhaps only a given sex on a given diet. So 10% of the 40 QTLs we found, 4 of them, were there in the whole population. If you had different diets and you had the different sexes, and instead of identifying them and analyzing them, you pooled all your data together to get the most out of it, most biggest sample size out of it, you would only pick up 10% of what's going on. This is a locus that shows such an additive effect, and that's not showing up great, but hey, that's life. I want you to ignore the 2 different heterozygotes for now. Dr. Wolf will be speaking about imprinting, and that's why we have 2 different heterozygotes here. Just collapse them for now. Don't worry about that. But you can see this is one that's over the entire population. There's no significant interaction. You can see the small genotype is fatter than the large genotype. And if you look at each category, the low-fat, high-fat, low-fat female, high-fat female, low-fat male, high-fat male, and you just look at the green and unfortunately the black, there and there, there and there, there and there, and there and there. You always see each, no matter which cohort you're talking about, you always have the same effect. The small is fatter, small homozygote fatter than the large homozygote. Now we can also look, so that's our original 10% in the whole population, and we can look at results that are specific only to the high-fat diet or specific only to the low-fat diet. These are situations in which there's a genotype by diet interaction, where the effect of the genes are different on the high-fat diet than they are on the low-fat diet. And you can see there's 10% of the results are specific to the high-fat diet, and 2% of them are specific to the low-fat diet. We go further and also look at loci that have effects on obesity specific to females and males. You see 15% are specific to females and 7% are specific to males, regardless of their diet. And then to break it down fully, now we can look at these 4 categories here: low-fat fed females, high-fat fed females, low-fat fed males, high-fat fed males. You can see that at 5%, I'm doing this right.
This one is low fat.
I'm going colorblind at the moment.
No,
These are the high-fat fed females.
Yeah.
44% of the loci only affected obesity in females fed a high-fat diet. They didn't affect obesity in the males on either diet or females on the low-fat diet.
5% is low fat.
5 fat is low fat female.
The 5% is low fat females.
Low-fat females, okay.
Yes.
And then the males are up here.
Yes.
So this is the high-fat males,
Low fat.
low-fat males and the high-fat males. So you can see that there's a lot of interaction with sex and diet, that the effect of the genes depend on the diet they're being fed and depend on the sex. Usually people do pool all these to make the most out of their sample. But if you have information on the sexes and you have information on diets, you need to take into account in the analysis, or you'll really get a false picture of what's going on. This is an example, again, ignore the, at least you can see all the bars on this one. Ignore the two heterozygotes for now. But you can see there's no difference in the low-fat fed males between the small and large homozygotes. Same for the low-fat fed females. High-fat fed males, no difference. The only difference is here in the high-fat fed females, where, again, the small allele produces more fat than the large allele. Actually, when you talk about body size, you know, the large is bigger than the small, certainly, but when you give the small a high-fat diet, it turns into a little tube of fat. And it's very highly responsive to the large high-fat diet. You do the necropsy, you cut the skin, and it blossoms for you. So this is that same locus we just looked at. It only had effects in the high-fat fed females, and that's shown here. Again, the high-fat fed females, small, fatter than large. The heterozygotes are kind of intermediate. So this is in the high-fat fed females only. This is in the whole population. You remember that a quarter of our population are the high-fat fed females, and they have a strong effect. That's 0.52. It means there's a gram difference between the small and the large homozygotes. But if we put that together and merge it in with the other three classes, the males and the low-fat fed females, we water down that gene effect so that when you pool over the whole population, you get nothing significant. So only one group has a significant result. If you looked in the whole population, you wouldn't see it. And since there are 4 equal-sized groups here, if you take that minus 0.52, which would be the 1.04 gram difference, divide it by 4, because only one of the 4 has 0.52, the others have zeros, you get minus 0.13. So you can easily hide, effects can be hiding if they're specific to a sex or a diet, and they can hide underneath total population structure. Here's just some more examples where we have this had an effect in females, but you can see an additive effect here at this locus. The large allele age fatter. But when you look in the high-fat fed females, there wasn't much difference between the large and small. Instead, the heterozygotes are all bigger. So there's dominance here and additivity there. So even though there is a gene effect in both categories here, the high and the low-fat fed females, the difference is changed. The genetic pattern is different depending on what diet they were on. Here's another example where we have high-fat fed females compared to males. And again, you see the massive difference with small being higher fat, leading to more fat than large, right there. Whereas in the males, it's really about equal, or if anything, opposite. When we looked at our loci, 80% of them had additive effects, 60% of them had dominance effects, and another 55% or so had imprinting effects that Jason will talk about later. So here are a plot of the additive effects. This is on a raw scale, positive and negative. So a negative number here means that small makes you fatter. A positive number, large makes you fatter. That's just by convention. You know, it could be the other way around, it wouldn't be a problem. This is the absolute value of these. So here, no matter which strain is making you bigger, it's all flipped over onto itself. So you can see this negative exponential pattern in the gene effect. So there are probably many genes of smaller effect we did not pick up. The average size of the additive effects is given here in grams instead of standard deviations. You can see that the average difference between small and large homozygotes for the reproductive fat pad is 1.2 grams. So to give you an idea, you know, in the mouse, the average size is 32 or something like that. So 1.2, that's a fairly big difference. You see the differences get smaller for the inguinal, mesenteric, and renal. And again, those also are smaller fat pads. The dominance, positive value is large dominant, negative value is small dominant. And you can see that they're about co-equal. When you have dominance, the large is dominant sometimes, the small is dominant other times. There's no overall pattern. You can see in terms of standard deviations that these values are similar to the size of the additive values. This is a look at the different kinds of dominance. Half of the loci are codominant, which is another way of saying there is no dominance. The heterozygote's right between the two homozygotes. So almost 50% that way. 14% of the loci are large dominant, and 6% of the loci are small dominant. 19% of the QTLs, though, the dominance values were over-dominant, where the heterozygote's bigger than either homozygote. And another 15% were under-dominant, where the heterozygote's smaller than either homozygote. One of the reasons that we did this in an F16, we've studied the F34, we now have data on an F44, and it goes on, is that you can limit the size of those confidence regions, smaller and smaller parts of the genome. And this gives the size of the QTL support intervals here in the F16 in terms of megabases. And you can see that there's really a peak here. The typical result is 2 or 3 megabases. When we were in the F2, it would have been more like 20 megabases. So we've got a dramatic reduction in the size of the intervals. Some intervals are long. That could be because there's more than one gene there. They're not far enough apart for us to detect yet, but they spread their effects over the chromosome more. And if we looked in these 3.1 megabase intervals on average, there were 27 genes typically in those intervals. In the F2, there would have been 200 to 400 genes. So once you're down to 27, it's not so bad. When we do the F34, which has double the resolution of this generation, then you're having more like 5 to 15 genes. And if you look in these intervals, you've got all sorts of great obesity gene names residing in these intervals. PPAR gamma, which is important for obesity and diabetes. A lot of drugs target PPAR gamma. ALOX 5. Carboxypeptidase E is the fat rat mutant. The fat rat is a mutant at that locus. Uncoupling protein 1. This uncouples mitochondrial energy production and from making the ATP. So it just burns without production of energy. IGF binding protein 4. IGF binding protein binds IGF-1, which is an important growth factor. PPAR delta, glucagon-like peptide 1 receptor. Unfortunately, the problem is we could go on for a long time. I could show you lots of gene names besides these ones. We also study diabetes in these animals. So this is a glucose tolerance test result. At 0 time, you inject glucose into the abdomen of the mouse. after first measuring the amount of glucose in the blood, they then do the injection, and you then measure blood 15 minutes, 30 minutes, 60 minutes, and 120 minutes after injection. So you can see since you've pumped glucose in, the sugar levels in the blood go up, but then they come back down again to normal in about 2 hours. And you can see that here with our low-fat fed females behave pretty well that way. They 120 minutes, they're back down to where they started. The low-fat fed males show a higher curve, and therefore are more diabetic, if you want to use that terminology. But again, you can see that it comes back down almost all the way to where it started. You feed the females high fat, and you can see that they, these are the low-fat males with the circles. The high-fat females are more diabetic than the low-fat females, but it's not a very big difference. While the high-fat fed males are really diabetic, they're coming nowhere near the starting point after 2 hours. So basically, we had the same structure here for diabetes-related traits that we had for the obesity traits, except things concentrated in the males, not the females. So of the interactions with sex, so results for this that were specific to a sex, 78% of the specific results were for males, 22% for females. And the high-fat diet is 63% versus 37% on the low-fat diet. And then if you break it into the categories, 72% of our QTLs were restricted to high-fat fed males, 28% the others. So on obesity, it was the high-fat fed females that got fat. Here it's the high-fat fed males that do. Again, a big sex difference in the genetic basis for the traits. When you put all those together, 64 diabetes QTLs and 40 obesity ones, 13 of them are in common, are in the same place. So I'll talk just for a little bit now about one single result. And that's shown here. It's on chromosome 1, and it's this QTL right here. You can see a strong effect for cholesterol level. A smaller but still significant effect of the reproductive or gonadal fat pad. A weaker result than that for free fatty acid level in the serum. And then the total fat pad just misses the threshold here. So if we're to look now at this position, and in that position there are 47 genes. It's a gene-rich region, actually. The list of genes here, of which there might be 10 or so, all are known to be related to obesity or diabetes or cholesterol level metabolic syndrome features. So there's a lot of good genes in there. Again, ignore the double heterozygote, but you can see that there's a really big effect where the large allele produces higher cholesterol levels than the small allele. And this is true for each part of the population. For the free fatty acids, the results are different in each of the cohorts. There's an interaction with sex and with diet. High-fat fed females, you see that same pattern we saw for cholesterol. Same pattern here for the low-fat fed females. We have, and for the high-fat fed males, but not for the low-fat fed males. We also have the total fat pad weight. And you can see here, there's no significant difference in the high-fat fed females for total fat pad weight, but there is in the high-fat fed males. And this one is one where there's no difference between the homozygotes at all, and all the differences involved in imprinting. So you go to that region of the genome, you go to the genome browser, and you get a really fuzzy version of what genes are in there. And the ones that have red boxes around them are reasonably good genes for cholesterol level or obesity. So this would be like the 47 genes, and then some of them highlighted. One of the ones that's highlighted here, oh, that's really interesting. I want to go backwards. Okay, is this one right here, ApoA2. Apolipoprotein A2. It's involved in cholesterol transport and metabolism. So here you can look, there's a difference in gene expression at that locus in the liver. We looked at gene expression levels for large and small on the high-fat diet and the low-fat diet, and this is actually across both diets. You'll see there's a higher expression of ApoA2 in the large strain than there is in the small strain. Many of the other genes that are potential ones in there don't show any expression difference. This shows, we have the full genome sequence of large and small, so we can tell where all the differences between large and small are. And this is a list of the coding difference, the non-synonymous coding differences in the whole interval. The R indicates that it has a potentially important physiological effect on the structure of the protein. NR is not so. There are three different criteria that are being used. They're polarity and volume, charge and aromaticity, and charge and polarity. So you can see that there are many possible ones here. The last column is mammalian conservation. So a high value there near 1 means that it's always the same base in this position no matter what placental mammal you look at. So ApoA2 has two such changes. One of them has no mammalian conservation at all, and therefore you'd think is unlikely to be important functionally. This one is a moderate level of conservation, so it could go either way. So this mutation has been picked out, the alanine to valine transition, as one that's likely to be responsible for that QTL by other workers. So we go from the quantitative trait locus here to quantitative trait gene, ApoA2, down to quantitative trait nucleotide, the specific alanine difference here. If you take a bunch of strains of mice and measure their cholesterol levels, you will find that those carrying the alanine have a low value for cholesterol level. Those carrying the valine have a high value. And you can see that in males and in females. Now, with our full genome sequence, we were able to see this difference, certainly. There are 6 other differences in mouse strains that are in complete ligacious equilibrium with this location. So that means those 6, if we did the same test at each one of the 6, we would get the same result because they're fully correlated in their distribution through the mouse strains. One of those is in a promoter region for ApoA2 and could be responsible, and it is highly conserved in mammals, could be responsible for this expression difference. We wouldn't expect the protein difference there to cause this. Now, it's possible that both are true, that there's one affecting expression level of ApoA2, and there's a second part of it affecting the protein structure, and that both are differences that are in inbred mouse strains travel together. They've been inherited together through time. To try to ferret out which of these is responsible, functional testing has to be done. And I'm not going to do that. But I have colleagues and collaborators who do those sorts of things. What you have to do is find some molecular geneticist who, for some strange reason, might be interested in ApoA2. People are interested in the strangest things. We got a new guy moving in the lab next to ours who's a protein biochemist, and I've been told that the stranger the protein, the more he likes it. So we're going to hit him up. Everybody wants to study ApoE, ApoC, not ApoA2 because it hasn't shown up as important in human populations so much. So that I think should fill the time for now. I don't think I have to go on anymore. I got lots. I took a lot of slides out. So please, if there's any questions, you ask them. Yeah.
I don't know if I'm recalling it well, but at the beginning you showed that the small strain, the females from the small strain are the ones that get fatter, right? Mm-hmm. Okay. So then you showed some other results, like for glucose, and it was the males
right.
that actually
from the small strain
have more tendency to be diabetic. I don't know. So isn't that a little hard to go together?
How do those go together? We'll talk more about this in another segment. But you're talking about, typically there's a correlation between obesity and diabetes. Mm-hmm. So more obesity, more diabetes. But in humans, that's also very different for the sexes. And males are more susceptible to type 2 diabetes than females in human populations, not to the extent that was shown here. This is extreme. Those correlations will break down depending on what your genotype is. In fact, if you look in these mice, there's not a strong correlation between obesity and diabetes. We're not quite sure why. One of the possibilities is there are different stages to diabetes. You start out, if you, I can draw on the board. If I do a graph and I got the x-axis is glucose and the y-axis is insulin level. And then you see small insulin, low glucose, that's the normal situation. Then you'll find animals that are low glucose but high insulin. That means their tissues have become insulin resistant and they have to pump out more insulin to keep the glucose as low as it should be. That's pre-diabetes. And those animals are heavier than the normal ones. You go to the third stage, which is high glucose and high insulin level. Now you're pumping out as much insulin as you can get, but it's not driving the glucose down. That's diabetes. The fourth stage is no insulin, high glucose, and death. As they pass from that second to the third stage, when one is diabetic, in an uncontrolled sense, you lose weight because the sugars can't go into your fat. It's resistant to them. So instead the sugar stays in the blood and the blood sugars rise. So what we actually saw was kind of a humped distribution where the normal group was a low weight, the pre-diabetic groups were heavy, but the full diabetic groups were back down to almost a normal weight, but probably due to a pathological cause. So the relationship's not entirely linear. Usually when they're talking about it in people, they're talking about people who aren't full diabetic. So that it's that pre-diabetes stage where the obesity and diabetes go together. Does that sound like an excuse I made up? It's rather involved. But it is actually the distribution of things in these mice.
And just one more thing. In nature, do you think a high-fat diet would be good for some kind of mammals? I mean,
I think it's a preferred diet by most vertebrates, even birds too. Oil in the seeds and stuff like that.
Do you think it would increase fitness?
Would it increase fitness in the wild? Not if you got to be an 85-gram mouse. She couldn't get through the holes very well, right? But within reason, it probably would improve reproductive success.
However, excess obesity will lower ovulation rate, both the time and number of eggs ovulated will go down when you reach an extremely obese stage. So, certainly there would be, if you want stabilizing selection at some intermediate level, because if it's fully, no fat at all, the reproductive rate's going to be low. The fat is used as stores, and what you see the pregnant female weighs a lot because of the pups, but she also has big fat pads. And then when she starts lactating, those fat pads shrink all the way down and are hardly there at all right after she weans. So certainly that energy is being used and put into the pups. It would be advantageous from that perspective.
Maybe that is related to why females, the genes for the females respond better in high-fat diets.
It could be. It could be. I guess the thing to say, one of the lessons to take out of all that data is that gene effects are conditional. They vary depending on sex. They vary depending on diet. I can't tell you whether this gene makes you fatter. You have to tell me more. You have to tell me what your diet is. You have to tell me what your gender is. Then I could say whether that gene would make you fatter. So, and these gene effects all do evolve. We'll talk more about that in the later times. So gene effects are not a property of the gene. They're something that has evolved through time. So don't think of a gene effect as being something that belongs to the gene. It belongs to the gene in its background, in its diet, in its environment, all those sorts of things. Yeah?
Do you have any suggestion if this works for humans the same way as human mice?
Well, humans are just terrible to work with. For one thing, I can control the breeding of these mice. And that F2 experiment we do controls it so that each allele is at 50% frequency, which gives you your best power for mapping. Humans have rare alleles. And I've yet to be able to convince at least half of the human race to breed the way I want.
Not even your kids.
People also lie. If you ask them about their diet, they lie. These mice had no opportunity to lie. So oftentimes in human studies, they don't have dietary information at all. And here we saw what a big difference that made. They do have the information on gender, and they'll have mixed ages. I had set ages where things happen. There'll be mixed ages in a human study. But and I had a nice balanced design of my diet, so I had all things going in my favor statistically. So you don't see many dietary studies. You very, very rarely see things separated by sex, even though it's, of course, well known that there are many physiological differences between males and females. But that's rarely taken into account. It used to be in mouse genetics that you wouldn't even study females. What would be the point in that? Every month they're up, they're down, they're up. You can't depend on a physiological state being more variable in females over time than it is for males. That argument actually never really held water. Isn't used so much anymore. But in human studies, it's still common to combine your males and your females to get your sample size up. And by doing so, if there really are diet interactions and gender interactions, then you are going to mask your gene effects. If there aren't, then you're going to get more power. You don't know unless you do the study. These mouse studies would suggest you better consider sexes separately and take diet into account. Any other questions? Okay. Thank you very much.